Welcome back to another episode of Ask the Expert, the podcast where we explore the science of bleeding disorders and treatments that are changing lives. I am your host, Amy Board, and today we are looking at the future flow of hemostasis research, how new discoveries are shaping therapies and improving care for people with bleeding disorders. This season, we've learned that hemostasis is all about balance, a system where the scale of procoagulants and anticoagulants can't tip too far in either direction. Think of it like a river system, a network of pathways, streams, and small eddies, those pockets of water that pause at the river's edge instead of flowing straight through. Each new discovery is like opening a new stream, guiding the current in more precise ways. From genetic variants to non-factor therapies, researchers are mapping the river in ways that were unimaginable just a few years ago. We're about to explore the flow of cutting-edge therapies, from factor-specific tweaks to rebalancing agents and RNA therapies with some of our favorite experts, Dr. Patrick Ellsworth, Dr. Janice Staber, and Dr. Ben Samelson-Jones. But first, let's hear from our sponsor.
Mark lives with hemophilia A. Along with spending time with his wife and their twin daughters, he enjoys volunteering and cooking. What you don't know about Mark is the impact that living with hemophilia has on his daily life. Mark bravely shares how hemophilia has impacted his relationships. He emphasizes that shared decision-making with healthcare providers, along with strong family and caregiver support, is key to managing the challenges of hemophilia. Scan the QR code or click the link in the show notes to hear Mark's full story and learn more about how important shared decision-making is for hemophilia patients and their support system.
Thank you again to Sanofi for their support of this podcast. We could not do it without you. So let's get into it. Let's rewind a bit back to the basics, the coagulation cascade, and how patient participation has helped chart this whole river of treatment.
Well, we're still learning a lot. And I think, all these explanations that we've given today, constantly evolve and there's nuances that we're learning about every year. But one of the ways that the initial coagulation cascade was mapped out was actually because of the participation of people with inherited bleeding disorders that were missing certain clotting factors. And they, working with blood scientists, they said, your blood is not working because it's missing this thing. And over time, enough patients with each disorder were identified, and some very smart scientists put it all together. And that's why they're all numbered kind of in the right order, but not exactly. And so, I think patient participation in blood science has been essential to understand this process, unequivocally essential. And I think it's still ongoing. And so we are understanding more and more about inherited bleeding and clotting disorders. We have not historically screened patients for tissue factor deficiencies, partly because, as mentioned, the tissue factor is not floating around in the blood. So hematologists didn't have access to that, but we now know that there are genetic variants in tissue factor that can decrease your ability to kick off the coagulation cascade. And that was published, I think, for the first time in just a few years ago. We're learning about new variants that can be dysfunctional in different ways.
So patients continue to help map the river. Each missing factor was like a missing stream. But understanding where the water didn't flow, researchers pieced together the system. And now that collaboration continues to guide how we understand bleeding disorders today.
From my nerd perspective, I'm going to highlight some of the work we're doing in the lab. One of the things we are interested in is understanding the diversity of how factor IX, the factor that's low in hemophilia B, can get messed up. And so what we've mostly done in the clinic is say, your factor IX is not working. You're at 2%. You have moderate hemophilia B. But now it can get messed up in different ways. It could get messed up because it doesn't interact with factor 8. It could get messed up because it doesn't interact with factor 10. It could get messed up because it binds antithrombin too good. I'm interested in thinking about those nuances and the different ways that you can mess up factor IX and thinking about if those have clinical implications for picking different therapies. And so, for example, we showed recently that there are some factor IX variants, protein, that cause hemophilia B. And one of the reasons they cause hemophilia B is because they don't interact with factor 8. But guess what? They interact just fine with emicizumab. And when you put emicizumab next to them, they can do not exactly like wild type factor 9, but pretty good doing what it's supposed to do.
It's like a river forming a new eddy around a rock, a swirl that redirects the current in a similar way. These molecular insights are creating new flows for therapy, like imicizumab, helping us navigate around traditional pathways. That brings us to how researchers are rethinking treatment altogether.
Traditionally, we think about replacing whatever you're missing. So for factor 8, that would be replacing factor 8. For factor 9, replacing factor 9. And those therapies have been around for decades. It came about, 10 plus years ago now, more than that probably, where the thought was, okay, well, is there something that we can do that is different from that, especially for inhibitor patients, because replacement doesn't work for inhibitor patients. So is there something that we can do that either mimics factor 8, and that is a product called Hemlibra that's on the market, or imicizumab? But then it was like, okay, well, maybe if that doesn't work, maybe there's other thoughts. And those you alluded to are called rebalancing. And so that means if somebody has a bleeding problem, if you give them, something that leans more towards clotting, could we balance that out? Could we make it so that then you just don't have an issue bleeding or clotting? It just is kind of the teeter-totter, if you will, if some of those diagrams are out there, has come and it's all equal. It's not down on one side. It's just balanced. So hence the name, these rebalancing agents.
Dr. Staber compares hemostasis to a teeter-totter, tipping the system just enough so clotting and bleeding stay in balance. Rebalancing therapies act like new currents in the river, creating pathways where clotting can form when it's needed. Within these flows, proteins act like cofactors, along with the cells lining our blood vessels and guiding the currents, shaping how the clots form and keeping the system in harmony.
But coagulation proteins are all these proteins, and there's different types. There's ones called proteases, which are kind of like scissors. They go around and, kind of cut things and do things, and they change other proteins. And then there's things that are called cofactors in the coagulation cascade. And there's all sorts of those proteins. And so for hemophilia, the ones we hear about the most are factor 8 and factor 9. And I'm interested in how those interact with endothelium, which is the cells that line all of our blood vessels, because it's a huge surface area inside our body that we don't usually think of as being involved in coagulation, but is important for basically putting those proteins where they need to be for hemostasis and thrombosis, and also for regulating that process.
Rebalancing agents and RNA therapies are both ways to help the clotting system, but they work differently. Rebalancing agents tweak the system's natural balance, nudging clotting in the right direction without replacing those missing clotting factors. But RNA therapies go a little deeper. They change how the body makes certain proteins, adjusting the flow of clotting from the source. Now, both approaches open new paths for treatment, but each takes a unique route.
And so what does that look like practically? That is taking a molecule that's known to affect the clotting side. So we know that factor 8 and factor 9 affect the bleeding. If you take them away, they can give you bleeding problems. If we change something that is maybe known to lean towards a clotting problem, could it offset that? So one of those molecules is TFPI. And so if you block that, or anti-TFPI, could we rebalance that? So could there be less of the things that then would inhibit then forming a clot too? So there's a couple of those that are new agents, and both are FDA approved, out on the market right now. The other strategy, though, is not to the TFPI, to block the TFPI, but it's to block another molecule called antithrombin. So antithrombin also inhibits thrombin, which then ultimately thrombin helps make fibrin, which is ultimately in how I describe it to patients is the kind of the mortar of the bricks and mortar system. So if you think of that your blood vessel would be like a round tunnel that is made of bricks and mortar, that if you put a hole in a brick wall, you need bricks and you need mortar.
Platelets are the bricks, and fibrin is the mortar. And each therapy is like a different kind of builder, patching the wall wherever bleeding threatens, each in its own unique way.
So the platelets are like the bricks, and the mortar is kind of everything else. And so the fibrin is the end game. That's the mortar. That's the big part of the mortar. So thrombin helps make it into that nice hard mortar. So we need really good mortar. If you block thrombin, so antithrombin, so if you block thrombin, then you don't get as much of the fibrin or that mortar part. So we're going to block the blocker. So if we can stop the antithrombin, then the whole thing can kind of proceed. And then we get the nice strong mortar at the end. So the mechanism there is to just inhibit the production from the beginning. So there's a new technology or not new, it's new to our field, technology that inhibits RNA. So molecules get made from like the DNA to RNA to protein. So if you just block the RNA, you can't get protein or as much protein. So that is the siRNA platform that inhibits the antithrombin. So that molecule is also out on the market. And so that one's Qfitlia. We all are getting used to all of these names, but it's the technology is the siRNA to antithrombin. So that's also a rebalancing agent because it helps, with that teeter-totter. So both of those strategies are kind of blocking the blockers to get the mortar, right? So we want to get the mortar. So if we kind of just let it flow and kind of stop blocking, that's the strategies for both of those.
So rather than adding more materials to the wall, these therapies focus on stopping what weakens it, restoring balance by blocking the blockers.
They have preclinical data showing, I know it's VGA039, I think is the one. And that one's an antibody to protein S. And so like I'd said earlier, a lot of these coagulation proteins, they have the protease, the scissors, the chopper, and then a cofactor. And so protein S is the cofactor for activated protein C. And without the protein S to help it out, kind of like factor 9A, activated protein C isn't very good at its job. It really needs its hand held by protein S. And so the VGA molecule, it's an antibody to it that prevents that interaction. So prevents it from basically fulfilling its function, the activated protein C from doing its thing. And so STAR is looking at von Willebrandt's right now because any of these potentially could be used as what we would call kind of a universal hemostatic agent, any of these rebalancing therapies. So they could end up having promise for like factor 7 deficiency, factor 11 deficiency, things like that, if it's particularly severe. Whether or not we want to use them in that is kind of a different conversation, but there's that potential. And so that VGA one from STAR is looking at that.
Rebalancing therapies guide the clotting system like new channels in a river, letting it flow where it's needed. Some block natural inhibitors, others create a pathway for clotting to form more efficiently. And the result? Patients who don't respond to the standard factor replacement can still see the system tip back into balance.
And then there is a protein C one. There's one that I actually don't know if they're still in. Last I heard, they were kind of like phase 1, like very early stuff, called Serpin PC. And that is a molecule that it's basically a decoy for activated protein C's target, which is 5A. So protein C usually is going to go to 5A and say, like, you're done here. I'm chopping you up, like we're done making thrombin or done inhibiting, thrombin from being made. So it acts as sort of this decoy and with some fancy biochemistry keeps protein C from inactivating 8 and 5. And so it enhances thrombin generation that way. And that one, for a lot of reasons, is just kind of biochemistry type reasons, is a really cool mechanism too, and could potentially be useful for hemophilia A and B and, other factor deficiencies and that. Those are probably the furthest along in terms of non-factor therapies. And there's a Chinese group looking right now actually at a non-factor, not a rebalancing, but a non-factor therapy for treating bleeding. So not for prophylaxis, but to treat bleeding. And that one is by directly activating factor 10, which is incidentally also how a lot of snake venoms work. So it's kind of a risky proposition, but kudos to them. They presented some data at ASH, some early stuff in hemophilia A and B with inhibitors. So that's another thing in the pipeline.
From antibodies to RNA, the field is creating new currents that steer the system towards clotting. Dr. Stabler shows how those discoveries move from the lab to real patients.
I think when these technologies came out, some of them, technologies and manipulating some of the antibodies. Other fields have used those for a long time. So like the siRNAs, we've used that in the lab to knock things down in the lab to test function for a while. So, when that came out, it's a really awesome technology. And then to see it applied to actual treatment. So most of the time we use that anti-RNA. So that's what it is. We're like blocking the RNA production. We use that in the bench lab, like in, just right outside my door here in the bench lab. We use that a lot to test to see if you block a protein or block an RNA, what effect that will have. And that's kind of where these got started, was in a lab that tested out blocking RNAs, and is that possible, and are they stable? And so just to have seen that as we use that for lots of different molecules in the lab to, now it's going to be a therapeutic. That's pretty amazing. And the fact when I started med school, it was kind of, it's going to be in 5 to 10 years. And then when I went from, med school to residency to fellowship, it was also like, 5 to 10 years. And I was like, I thought that was going to be the mantra for the rest of my life. In 5 to 10 years. And so for that to actually be in our tool belt as a potential for patients, especially again, for individualized for patients who just want to break from infusing, to having a hard time with their infusions, to, whatever the reason for those individual patients. The fact that it's actually been effective enough to make it to market has been pretty remarkable, just given the journey and the hurdles that have had to overcome in that field. That's been pretty remarkable.
We've come such a long way, from rebalancing agents to RNA therapies. And at the end of the day, these aren't just molecular discoveries. They're tools clinicians can use to guide real people through their unique treatment journeys.
I'm interested in thinking about this both for emicizumab and other factor mimetics, but also thinking about other non-factor therapies and how does that affect how we should be counseling patients, and really drilling down to the molecular interactions that underline the coagulation cascade in order to better advise patients about what might be most helpful for them.
By studying how different variants respond, we're finding calmer eddies and safer currents in treatment. Dr. Patrick Ellsworth has been thinking a lot about what this means for patients who still need breakthrough bleed management, especially those with inhibitors.
Yeah, so last nerdy thing, and what I'm interested in from a research standpoint and hope, and really am interested in with these folks, is breakthrough bleed and surgical management for people, especially who still have inhibitors, and what we do with those, because I'm not seeing little kids anymore, but a lot of my pediatric colleagues are finding that kids on emicizumab with inhibitors, you'd start talking about ITI and they're like, no, thanks, we're doing fine, because they're like, kid's not bleeding anymore. We're on emicizumab. Like, why would we do that? So I'm interested in okay, what do we do with those folks? And if they need surgery and they're bleeding, they still have an inhibitor, what do we do? Because right now we're really just stuck with activated factor 7. We've got a couple different brands of it, but that's about as much of an option as it sounds like, which is that it's, different flavors of the same thing. And it doesn't always work. Sometimes activated factor 7 doesn't work, and sometimes people aren't comfortable using it. It's very pharmacy and labor intensive to give someone that. And so I'm interested in looking at other ways that we can kind of leverage the unique mechanisms of these things to improve breakthrough bleed management on these non-factor therapies and how they act differently.
#MyHemophilia_Truth. Real stories powered by Sanofi. It's time to consider the whole experience when it comes to managing hemophilia and change the conversation. Explore the full story at www.myhemophiliatruth.com.
So we're branching into new channels, reshaping how care is delivered, and revealing what's next for bleeding disorders. Next episode, we are going to recap the season's top insights by showing how far the river has traveled and where it's heading next. Until then, keep exploring, ask questions, and stay ahead, because the future of bleeding disorder care is truly flowing forward. Did you know bloodstreammedia.com is your one-stop shop for all podcasts for the bleeding disorders and rare disease community? There, you can find links to the Global Hemophilia Report and our flagship podcast, Bloodstream, with your hosts, me, and Patrick James Lynch. Also check out Flow, which is straight talk about extreme periods, and The Pain Pod, which is an in-depth storytelling odyssey of chronic pain. All this and more on bloodstreammedia.com, the #1 podcast network for rare disease. Have an idea for a future episode of Ask the Expert? Or have a question that you would like to hear one of our experts answer? Email us at mailbag@bloodstreammedia.com. You'll also find Bloodstream Media on Facebook, and on Twitter and on Instagram and all those social media channels. I am your host, Amy Board, and until next time, stay curious, my friends.